Do you know why hotel sheets are much longer than your domestic sheets? Hotels used to change sheets but not blankets, and there was a worry that somebody that had tuberculosis might have left their saliva, their sputum or coughed on the blanket. So these hotels had a very long sheet so that you weren't worried about who'd slept there the night before and whether they had TB.
We are in the Diagnostic Microbiology Laboratory of Addenbrooke's Hospital in Cambridge, where we accept around 800,000 specimens from patients with suspected infectious diseases every year. They are logged in and barcoded, and they then enter into the diagnostic laboratory where they go through a workup pathway. Our job is to detect pathogens that may be causing their infections, to identify them and to determine what antibiotics to treat the patient with.
Germ theory is the idea that microorganisms cause human disease. But that sounds more simple than the history actually was. It took quite a long time for there to be closure on the idea that tiny organisms had the power to cause major changes in the human body and even cause death. Initially, the germs were thought to be chemicals, and the word virus comes from the word poison. It was really Pasteur's work on fermentation, which he went into as a chemist. He began investigating fermentation, and he discovered that it was actually being caused by living organisms, yeast. That was particularly important in France with the fermentation of wine. But it also changed the way that doctors thought about infectious diseases.
Over the course of my career, the way that diagnostic microbiology has changed is that we've taken techniques that were developed 100 or more years ago, such as microscopy and growing bacteria on agar plates. We've mechanised that to make it much more efficient to do. The other important technological advance in my lifetime has been the introduction of molecular tests. Whole genome sequencing of pathogens means that we can actually sequence the entire genome of the bacterium, for example, a sample that comes through the door from a patient specimen, and we can understand in great detail about the genetic repertoire of that particular pathogen.
In terms of medicine, it meant that doctors increasingly focused their attention on the agent of disease. I mean, we now know that there are bacteria, there are viruses, there are prions, a whole set of germs. We are broadly kind of probably too complacent about it, but there are worries with antibiotic resistance, with the spread of new diseases from abroad, and on a global perspective, infectious diseases are still a major cause of death and globally far more important in proportion than they are here. Any breakthroughs in the future are going to be at the fundamental molecular biochemical level.
I believe that whole genome sequencing will move from the research environment where it's placed at the moment into a diagnostic laboratory setting where genome sequencing could actually be used on a daily basis to guide patient management, to investigate outbreaks and improve our control of infectious diseases generally. One of the major challenges that we face as treating doctors is the ever-increasing rise in resistance to antibiotics that we see for important human pathogens. Now, we could use whole genome sequencing to have a better understanding of how these pathogens are being transmitted and therefore controlled. So this may be very important in future control of drug resistance. It has huge potential to improve individual patient care and to have control of infectious diseases both at a national and a global scale. That's what drives me, really.